Okay, this is the long version, the long video tutorial of how to do that. And you don't have to watch it. I'm just going to go step by step in case that you have a doubt about how to do something. So in the beginning, you just have to go and click here, or you can copy this and you will go to this website. And as I said here, you just go and look into the different tabs. So let's go together and do that. Here, for example, I can read about the learning objectives, how to use PCR, how to identify bacteria. Here you have information about the samples. Here it will be the different DNA sequence will appear here, how to use experiments. Here you will have your notebook and they will give you more information here, the reference, and here the help menu. So I'm sure that you already watched the video. And here let's start. So the first step is it says click here to enter to the lab. So you just have to wait a little bit. And now we have to do is read this and read this. So here, welcome to the virtual ID lab. And please put a pair of gloves. So by clicking on the glove box in the open drawer, so you go here, then you keep reading here. They said now you will obtain sample from the culture dish. That this is what you generally do in reality, and if this is what we hopefully when the pandemic is over you will do. So we take this and we click here. Now click on the bacterial colonies. These are the bacterial colonies, and this is what you do. So now you successfully obtain the bacteria. Now click on the microcentrifuge tube and to transfer the bacteria from the colony wire loop into the microcentrifuge tube. So here, and now placing the bacteria, you have successfully placed the bacteria. Click on the yellow micropipette. Then you go here and you are moving from one side to the other side. I recommend you to keep reading all this information. Oh look, here it already appeared, but right now we don't need to go here. Here they tell you the important parts of the sample preparation for the PCR, and click on the microsync to continue. Now you have to wait several times, and this is an experiment that we generally do in the genetics lab. If you take genetics, you will do this experiment with me. So after several hours of the enzymatic digestion, your sample is ready to click the microcentrifuge tube to hit inactive the digestive sim. So click in the microcentrifuge. So we click here. It moved to this place, and I can tell you more about what is happening here. Then click in this tube. Click in the centrifuge leak to continue. This is the same. Now this will heat up the DNA, and now we will move it here. So the centrifuge here it was heated. Here it spin, so it separates the DNA. Click in the microcentrifuge tube. So now you transfer the supernatant to the PCR tube. And this is exactly what you do when you're doing a real PCR. Then the sample preparation is complete. Click on the orange tray to continue the second part of the PCR amplification. So here is the orange tray. And as you can see, here is where you move to the second part of the protocol, the PCR amplification. Now you can read all this information so you learn about PCR. And I just tell you that you have to click here in the red cap and you pipette this into this. And here you can read more about the master mix solution. Now as I click the green cap, so now this continue. Then use the click the blue cap ionized water bottle. So let's click here, and now you will have to pipette this. Then it said click the PCR machine to open the loading. So now you're going to move this here. So you take all the samples and you put it into this machine that is called a thermocycler. And in the thermocycler, what you have is the change in the temperature. And during this step, they run the PCR, and now we have to click to close the machine. And CMU has a thermocycler, and here they explain what the thermocycler is doing. That is using the complementarity of the bases, is allowing the DNA to be copied, and we are making photocopies of the DNA to be able to have extra copies. So when we have all these photocopies of the DNA, we can and we get amplified all this sequence, we can easily identify the DNA, because instead of having a very small amount, now we have a lot of those copies, and we can sequence it much easier in that way. So now they're showing how many copies you're getting. So look, I start with 2, then I move to 8, and now I have millions and millions of copies. And now I can move to the next section, and here now I have to click on the orange tray and go to the PCR purification. Here you have to read this, and I have to click on the buffer solution, and this will pipette again, and we're going to add the buffer solution. Here they can tell you more about this, and a buffer solution, as we study in class, is a solution that allows you to maintain the pH constant. Now click here to continue, and you can continue pipetting. And then you have to put these samples in the centrifuge, and this will, you have to counterbalance them. So now we're almost done finishing the PCR purification, and this is exactly what you do in the lab. Now we get rid of the extra sections, collect the DNA into a collection tube, and we again go and put it in the centrifuge. We close the centrifuge. We wait a little bit more to separate the different solutions that we have here. And now we are going to the sequencing preparation, and you have to read this, and so you understand in more detail what is happening, and you can keep clicking to understand what are the other steps. And here, they will tell you all the steps again. Sometimes there are pipettes that instead of having one, they have multiple pipettes at the same time, so you only pipette one thing 6 times, and in that way you can save time. Click the PCR machine, so click here, we put everything here again, and now we're copying again PCR of all the samples. And now we have all this sequence. Now we go to the DNA sequencing, and please again read this part, and you can continue clicking in this section that it will give you all the information. Click the DNA sequence machine, so here load the trait, and here, as this DNA sequence, they have a special fluorescent. They will give a specific color, and you can see all the T's, A, C's, and G's. Before, this was done manually and took hours. Now a computer is doing this, and now we can have in a few hours these results.